Then the next slide is, as you can see here, is a yellow color structure. So it is bile stained. Then what is this? The questions. It is a saline mound of feces sample. Then identify the structure focused. You can see the structure focused is Ascaris lumbricoid fertilized egg. That is fertilized egg of Ascaris lumbricoid. So you can see this is the structure, as you can see here, fertilized egg of Ascaris lumbricoid. So the features of fertilized egg of Ascaris lumbricoid are, it is round or oval in shape, as you can see here, round and oval. Then bile stained yellow color, surrounded by thick, smooth, transparent shell with an outer albuminous coat which is thrown into rugositis. As you can see here, thick shells, transparent shell. And then contain very large unsegmented ovum with a clear crescentric space at each pole. As you can see here, there are clear crescentric space at each pole with the unsegmented ovum. Then floats in saturated solution of common salt. Then the next question was, what is the habitat for the adult worm? The habitat for adult worm is small intestine. Then the next slide is, as you can see, it is slightly different from what we saw earlier. So let's look what is this. This is a saline mound of feces sample. Identify the spotter and give 2 points for identification. So this is a egg of trichuris egg. So you can see it is bile stained and barrel shaped with mucus plugs at either pole. This is a clear picture. This is barrel shaped and mucus plugs at either pole. Then contains an unsegmented ovum. It also has unsegmented ovum. Then floats in saturated solution of common salt. This is egg of trichuris. Then the next one is, I can see here, we can easily identify this thing. What is this? This is rhizopus. This microscope shows rhizopus. So identify the fungus. It is rhizopus. What type of fungus is this? It is again mold and saprophytic fungus. Name 2 fungus causing zygomycosis. Zygomycosis are caused by rhizopus and mucor. You can see this is rhizopus. And the next slide is, it is a tube containing SDA with growth. Describe the growth in the tube. Okay. The growth is characteristics with cottony wool. As you can see, for the characteristic white cottony wool colonies with tube filling growth. The tube is fully filled with this growth. In rhizopus, it become brown black later due to sporulation. Rhizopus become brown black in later because of sporulation. Initially it is white. So name 2 fungi causing zygomycosis. Zygomycosis caused by rhizopus and mucor. Describe the growth in the tube as we already described it. And the next slide shows, this is, as you can see, the background is stained. So it is a negative staining. Okay. So name the staining method used. Is a negative staining. Name 2 capsulated bacteria. It is used to visualize the bacteria which are very difficult to stain. So the bacteria like Haemophilus influenzae, Neisseria meningitidis, Pseudomonas aeruginosa, etc. are stained using this method. Okay. And the next slide is not clear, but it is a gram staining showing gram positive cocci arranged in clusters and pairs with numerous pus cells. Gram positive cocci arranged in clusters and pairs with numerous cocci. So It is a staphylococcus. Then a gram smear of pus obtained by diabetic patient with a carbuncle is focused here. Then write down the probable identification of the organism. Why? Forward gram smear shows this shows gram positive cocci arranged in clusters and pairs with numerous pus cells. Difficult to identify. Then name two toxin-mediated diseases produced by pathogenic species of this organism. So it causes exfoliative toxins causes staphylococcal scalded skin syndrome. That is SSSS, staphylococcal scalded skin syndrome. Then that is exfoliative toxin. Okay. Then there is endotoxin, which causes staphylococcal food poisoning. Then, for example, and then other one is next question is about what is this? Identify the test. So you can see test I will show. This is the test. Identify the test instruments. Then identify the test. What is the purpose of this test? Is it epsilometer test or e-test? Okay, epsilometer test or e-test. Then write down one advantage and a disadvantage of this test. One advantage is it is easy to perform. Disadvantage is that it is very costly. Then it is not suitable for Cryptococcus neoformans. Okay. Yeah, it is easy to perform, but it is not suitable for Cryptococcus neoformans. Then you can see it has various layers. It is antibiotic strip placed in between. It is zone of inhibition, and then bacterial growth on agar plate, as you can see here. Then this scale is used to read MIC, minimum inhibitory concentration, where the edge of the inhibition ellipses intersect with the side of the tested strip. Okay guys, the next I will show in the next video, otherwise it will become very lengthy. Okay, if you like the video, please like.